Just
Hello.
shortly. Hello.
Sama.
Can you
I
see
can
my? Can you see the slides now?
Yes.
Okay, great. So we are working at my department with DNA barcoding.
Okay.
And DNA barcoding is we use. I heard you also use it in your research, right?
Yes,
So
sir. Yes, ma'am.
Oh yeah, that's great. So you use just a small sample to amplify a specific region of the DNA, and this region is very variable, so it can be used for species identification, and later for phylogenetic assessments and so on. This is what is called a DNA barcode. It's just because you can use it to differentiate species, just a fragment of the genetic region.
Okay.
And then you can use as material several different samples, just a small sample that you will collect from individual samples, and these will be amplified using a particular method, Sanger sequencing. And then, I don't know if you did such analysis, you can align the sequences and later on assign each of these sequences to sequences that are available in the database. There are some databases such as GenBank and CBI, where everybody who produces those sequences that are linked to a specific organism, we can use these sequences to compare with our sequences, and then you can identify the species just based on the molecular data.
Okay.
So this is also very important, for example, for monitoring of logging, since just a small material of, for example, a piece of wood can be used to amplify these barcoding regions, and then you can identify the species using just a little piece of material that you don't know, maybe you don't know the species, you don't know if this sample is from this wood, maybe it's from an endangered species. So you could use that to identify if this exploitation is from one endangered species. So here you can see how the workflow of DNA barcoding method happens. So first we have the sampling of your studied organism, maybe one specific plant, and then we will just have a little piece of the sample, just a little bit of leaf material, and these will be processed. This is by DNA isolation, where you can isolate the DNA and later on amplify using polymerase chain reaction, and then later on you can sequence that using Sanger sequence. I have another slide I will show. And after all, you can compare the sequences that you obtained in the lab with sequences that are available in the database, for example, barcode of life database and GenBank, for example, here you can see. And then you can assign your sample to these specific sequences from the database, so you can identify the species on a molecular basis.
Okay.
This is another. So the main point in the DNA barcoding is that the genetic variation inside the same species will be very small. So this is the intraspecific. The intraspecific, so the variation inside the same species will be very close to zero.
Okay.
And the variation between two different species or several different species will be very high, so that we can see that the species, you can really differentiate them. For example, inside the same species, you don't expect much variation for these barcode regions, but between different species, we expect a lot of variation. That is called this barcoding gap, when the variation between different species is much higher than the variation inside the same species. So this is a good marker, good genetic region when you find results like that. However, in some cases, the same barcode, it's not so efficient to differentiate between species for some specific taxa or some specific species. And then sometimes there are cases that the intra-specific variation, the variation inside the same species is so large, and the variation between two different species is very low, that the barcoding marker cannot be used to distinguish some species. So there is these problems also in the barcoding methods.
Okay.
And then the main aim of this DNA barcoding is to facilitate the morphological identification of plant species or other organisms using DNA. So for example, for a taxonomist, as you can see here, if you have a plant that you don't know what's the species of this specific plant, and then the taxonomist, the person who will identify these samples based on the morphological, on the shape of the leaves and all the characteristics and traits of those plants. So these, they take a lot of time to identify the species, right? Comparing to herbarium brochures, as you can see here. However, using the DNA barcoding, it can narrow down. Maybe, for example, this is species that you don't know the identification. You already know that belongs to the same tribe, and it's very closely related to these genes. So the expert that's identifying the plants can look only to those genera that is very closely related to this one, since they group it close together. Just another aspect, that there are several species that hasn't been discovered yet. So from all the species that are presented in the world, 80% of those species, they are still undiscovered. Nobody discovered those species. So imagine how long it will take until someone is expert to identify these new species. For like, nobody has yet studied them. And then you can see more than 8 million species are not yet described. So this method facilitates the identification of new species as well. So here I already talked about this slide. So some other applications. There are some species, for example, here in butterflies. These are two different species, but for me and maybe for you, we are not experts on butterfly identification, right? Yeah. So then you can see that we cannot really identify them. But with this molecular identification, the DNA barcoding, we can identify those species that are called cryptic species. They are species that they look exactly identical based on the morphology. However, they are different species. Maybe they occupy different habitats. They are different species, but they are maybe very closely related. And then this method can identify those species that they look very similar, but they are different species. Hello.
Hi.
Hello.
Hi.
So other applicability, for example, is the identification of some very immature forms. For example, eggs or fishes or mixed samples, or even for food authentication, since you just need a little bit of material for the DNA isolation. And also, for example, for mixed tea samples, sometimes we drink tea and we think, okay, I'm drinking jasmine tea, but actually maybe there is some other plants inside. So this method can be used for the for food authentication. So we are really drinking what we think we are consuming, right? So that's also very important. Other aspect is for the identification of disease vectors, for example. So you can also use the DNA barcoding to identify some disease vectors. And then, because even like mosquitoes, they are also important for the ecosystem, since it can be just food for other animals and also pollinators in some case, for example. So then it's still important to have them in the ecosystem, even though for us it's not so pleasant. But not all of them are vector of diseases. So when you use this DNA barcoding, so you can identify the species, you can also decide which are the right decisions for the management and to avoid some specific disease, so how to fight those. Other aspects also for identification of endangered species. So you can see here, for example, some birds and plants, and also the shark fin. Sometimes they are used and it is an illegal trade, and the state of this material is already so bad that you cannot really identify which are those animals and plants. So you can use just a little bit of this to identify the species.
You can share the document. I don't know.
Yeah, I will send the slides then. You can,
so that they can see it and also.
You can look. Yeah, I was showing. But anyways, I will have to go because I will have.
No problem. No problem. Thank you so much.
I hope it was some new information for your group. And I will send the slides so then you can check. Okay.
Thank you so much.
But I have told me before. I thought everybody was. Thank you a lot. It was a pleasure to talk to you.
Thank you so much.
Thank you. I talked to also see the presentation.
Thank you.
Thank you too.